Let's talk a little bit about osmosis. Osmosis is the movement or diffusion of water across the plasma membrane. Here's the deal though. We already said in the last video that water, being a small polar molecule, does not cross the plasma membrane directly on its own very well. So what you will eventually see as you survey the plasma membrane and diffusion a little bit better is that all the examples of different molecules that do not cross the plasma membrane very well, with the exception of large molecules like DNA and proteins, these molecules can cross the plasma membrane, they just can't do it on their own. They will need help in the form of channel proteins or carrier proteins. In the case of water, water being a polar molecule cannot cross the plasma membrane very well on its own. So what you're going to see here is that water cannot cross here, but what it can do is it can cross through a type of protein that spans the entire length of the plasma membrane right there, and this particular channel is called an aquaporin channel. Most of the plasma membranes in all the cells of your body will have these aquaporin channels, meaning that even though water cannot cross the plasma membrane directly on its own, it still gets to where it wants to go with relative ease because these channels exist almost everywhere and they exist in great abundance. So water, in spite of being constrained by its diffusion limits, can pass through the plasma membrane fairly easily, but it's only because of this aquaporin channel. So even though water can get from place to place relatively easily, water does not just move for the heck of it. Water will only move, being osmosis will only occur under a set of different conditions. There are actually 3 different conditions that all need to be met in order for osmosis to occur. The first one we've already discussed, so water has to be diffusible. If it was not for the aquaporin channels, then osmosis would not occur at a respectable rate at all. So in order for osmosis to occur, the particular plasma membrane or membrane in general that you're talking about has to have aquaporin channels, or it needs to have some way for water to get through. If water can't get through, it's not worth talking about osmosis because it's not going to occur. The second is that there has to be a gradient for different solute molecules, basically meaning there has to be a difference in the number of solute molecules on one side of the membrane versus the other. If you look at this example right here, we have these purple solute molecules, which we're going to use for our example here. When we say that there is a solute gradient, we're really referring to the fact that there are 10 solute molecules over here and only 2 over here, so there is an inequity. Basically, the driving force behind osmosis is that these molecules right here are cramped and they are going to want to get some elbow room. So these molecules over here are going to want to move from the left side to the right side, from side A to side B, so that instead of having 10 over here and 2 over here, they want to move until we reach equilibrium, being that there would then be 6 molecules on the left and 6 molecules on the right. The final condition, though, is that those solute molecules have to not be diffusible, essentially meaning that if these molecules were diffusible, there would be no reason for osmosis to occur. These molecules would be able to reach equilibrium on their own. If they're able to cross the membrane on their own, then they will take care of it on their own, and condition 2 would then disappear, meaning osmosis would not occur. So the 3 conditions: water has to be able to move, the solutes should not be able to move, and there should be a gradient of solute molecules on one side versus the other. So here's the other question. We know that the big driving force here is that the solute molecules want to move so that there's an equal number on both sides. But because these solute molecules cannot move, they cannot cross this membrane here, we know that that is not going to be possible. No matter what we do here, we are going to have 10 molecules over here and 2 molecules over here. So we need to think a little bit. If we know there's always going to be 10 here and 2 here, what can we do to give these guys over here a little bit of breathing room? So in a sense, the driving force behind osmosis isn't necessarily to get an equal number of molecules, It is to equalize the concentration of those solutes on one side versus the other. So when we talk about what the actual strict definition of concentration is, we're just talking about the number of solute particles per unit volume of solution. So if you look at our setup here, we have side A versus side B. So side A and side B are separated by a plasma membrane that has this aquaporin channel stuck in the middle of it. So in green here, I have also outlined what the volume of solution on side A and side B are. So in both cases, to start out with, each side has 1,000 milliliters of volume of solution on either side. So there's 1,000 liters of water, or 1,000 milliliters of water over here, 1,000 milliliters of water over here. There's 2 solute particles here and 10 solute particles over here. So if you're comparing the concentrations of both sides, we have 10 particles over here per 1,000 milliliters, 2 particles over here per 1,000 milliliters. So those two fractions are obviously very different. So we would say that side A is much more concentrated than side B. It's actually 5 times more concentrated. So the driving force behind osmosis isn't going to be to equalize the exact number of particles, it's going to be to get the concentrations to be the same. So rehashing our conditions for osmosis here, the solute particles have a gradient which they want to alleviate, but they cannot do it themselves because they can't cross this membrane. Water is going to do this for them because they are not able to. Water can't do anything about the number of solute particles in the concentration equation, but water can do something about the volume. So water is going to move from one side to another, not to change the number of solute particles, but to change the concentration by increasing and reducing the volume of solution on one side versus the other. So the driving force here is that the concentration in side A is too high and the concentration in side B is too low. What's going to happen here is that water from side B is going to move over to side A. So in this case, we would have water here not moving in this direction, but water would actually be moving from right to left, from side A, or excuse me, side B to side A. So water is going to move from the right side to the left side. So if you want a rule to live by, osmosis will always occur as long as these conditions are met from the side of less concentration to the side of more concentration. So what you're going to see here is that as water moves from side B to side A, the volume here is going to start to go down, whereas the volume over here is going to start to go up. So this volume will go down, this volume will go up, so that if you look at the equation here for calculating concentration, as the volume inside B goes down, the concentration will start to go up, because when you decrease the denominator there, you increase the value of the concentration. And then inside A, as you increase the volume because of all this water rushing into side A, you increase the value of the denominator, which decreases the value of concentration. So you bring the concentration down over here and increase it over here. So if you have side B, which is too low, side A is too high, we are going to have water move until those concentrations are about equal. So in this case, if we always know that we're going to have 2 molecules over here, 10 molecules over here, we can't do anything about the number of molecules, but we can decrease and increase the volumes on both sides until those concentrations are equal. So that is the essence of osmosis. We're not trying to do anything about the disparity in the number of particles. We're trying to do something so that we equalize the concentrations so that we then get better elbow room. As water moves from the right to the left side, these 10 molecules over here are going to have more elbow room because the volume is going to increase and they will basically have more room to roam. That is the essence of osmosis. The solutes cannot move, but the water does, so the water equalizes the concentrations and not the actual number of particles.